I am Amit. I am a graduate student of University of Texas, Rio Grande Valley in Department of Computer Science. My presentation is about the interplay between the O-GlcNAcylation and the phosphorylation in diabetic heart for ISMB 2022. The motivation is the proteomic based mass spectrometry has shown where the PTMs are in proteome. Using the proteome data and computational methods, we can figure out that the protein activity isn't being controlled properly in obesity. More and more evidence is showing that the way O-GlcNAcylation and the phosphorylation work together is important for many biological functions. Given how important both PTMs are to the way the heart contracts, we mapped and measured the O-GlcNAcylation and the phosphorylation of myofilaments in normal hearts and the hearts of mice with type 2 diabetes. Introduction: the heart disease is 2 to 4 times more likely to happen to a people with diabetes rather a non-diabetic one. Diabetic cardiomyopathy is a progressive problem with heart's diastolic and systolic functions that is caused by the lack of contractile force in the heart muscle. 60% of the diabetics have diabetic cardiomyopathy, but no medicine can stop it or change its course much. most research lack on the lack of contractile force in diabetic cardiomyopathy has focused on poor calcium handling or the release of activator calcium from the sarcoplasmic reticulum before systole. O-GlcNAcylation is a new player in the way the heart contracts. O-GlcNAcylation levels are inversely related to how sensitive myofilaments are to calcium. Considering the role of site-specific O-GlcNAcylation and the phosphorylation PTM in cardiac contractile function, it would be very interesting to find out how much the interaction happens at the myofilament level in diabetic hearts or diabetic hearts and a normal heart. The experimental data was generated from 3 wild type and 3 diabetic hearts were challenged with force frequency response, and 1 wild type and 1 diabetic hearts were further challenged with isoproterenol. For every sample, we calculated the phospho and O-GlcNAc and the protein expression. Quantifies 5,973 phosphorylated sites mapping to 2,347 proteins in phosphoproteomics data, 1,354 sites mapping to 361 unique protein in O-GlcNAcylation data. There are 183 sites mapping to 95 proteins that are common between phosphorylation and the O-GlcNAcylation, and we have 237 unique protein between phospho and O-GlcNAcylation. We perform statistical analysis of both PTM data, and we identify the sites that are statistically significant in phosphorylation and O-GlcNAcylation. The data on both phosphorylation and O-GlcNAcylation are normalized based on how much the protein is expressed. In this volcano plot, the x-axis shows the fold chains after log transmission, and the y-axis shows the p-value after the minus log transmission. Figure shows the place where the fold chains are greater than 2 and the p-value is less than 0.05. As you can see in the picture, the O-GlcNAcylation data doesn't show any important size, but that doesn't mean they aren't also significant from a biological point of view. We look into them in more detail in the later O-GlcNAcylation fold chains and the phosphofold chains. we can see a strong correlation of 0.048 between the average fold change of phosphodata and the O-GlcNAcylation data of the same residue sites. The 8 sample expression data, then we look into the relationship between the phosphorylation and O-GlcNAcylation sites that are same. As shown in the figure A, the correlation between phosphorylation and the O-GlcNAcylation is much more likely to be right than the correlation between any other two random pair that are shows in the blue of figure A. We also did the same analysis at the protein level. You can see in the figure B, where we found a significant positive correlation between the phosphorylation and O-GlcNAcylation of the protein sites. To figure out how important the distribution is, we compare to its pair of phosphocoagulate that are chosen in random. The random ones are shown in marked with yellow one, and the matched protein pairs have a positive correlation in the co-phosphorylation, co-O-GlcNAcylation, and phosphocoagulation correlation in compared to their permuted one. We also look into whether the proximity of sites on the same proteins affects the correlation of post-transitional modification. The figure shows that the distance between the sites, when the distance between the sites increases, the correlation coefficient decreases in all three cases, with an exception, and you can see in the figure C, where the distance between two residues is less than 2, the correlation of phosphorylation and O-GlcNAcylation is lower. In conclusion, we can see there is a strong correlation between the phosphorylation and the O-GlcNAcylation of the same protein sites. Close intraprotein residues have higher correlation with phosphorylation and O-GlcNAcylation than those far apart. This work was supported by the two NIH awards. You can see the name here, and thank you.